Okay, so today we're diving into research that could change how we treat blindness. Imagine like damaged corneas being regenerated and sight restored.
Yeah, it's a huge development for sure. We're talking about limbal stem cell deficiency. LSCD is the acronym, basically the cornea, that clear front part of the eye. It gets cloudy and scarred because the stem cells are damaged, and that obviously impacts vision.
And right now there aren't a ton of options for these patients, right? Transplants come with a risk of rejection, and other treatments don't always work. But this research is using something called iPSCs-induced pluripotent stem cells, and it's offering a lot of hope. You sent over a Lancet article that goes into detail about the first human trials of this technique.
Yep, and to really understand just how big of a deal this is, you've also included this 2024 review article. It's from Advances in Ophthalmology Practice and Research, and it explores stem cell use in ophthalmology as a whole. So let's break down the science behind this and explore the study's findings and talk about what this means for LSCD and other conditions in the future.
Awesome. So first, can you give us like a quick explanation of iPSCs? Like what are they? How do they work?
So imagine taking an adult cell, like a skin cell, and basically turning back time biologically, reverting it to a state where it can become any cell in the body, like a stem cell. That's the amazing thing about iPSC technology.
So it's kind of like giving these cells a second chance to become something new and maybe even life-changing. And in this case, scientists are transforming iPSCs into corneal epithelial cells, which are the cells damaged in LSCD.
Exactly. And a really cool thing about this is that we can create patient-specific iPSCs, meaning the risk of the immune system rejecting them is much lower, which is a problem with regular transplants.
That's incredible. Now, this Lancet study mentions this thing called the seam cultivation method. Can you explain what that is?
Yeah, so the seam cultivation method is groundbreaking. Imagine recreating the environment of a developing eye in a lab. That's basically what this method does, and it lets them efficiently produce corneal epithelial cells from iPSCs.
So they're essentially building a mini eye to make these specialized cells.
Wow. What about the patients in this study? Who were they?
So the trial focused on 4 patients. They all had LSCD in both eyes. The age range was 39 to 72, and the causes of their LSCD were all different, which shows that this treatment could work for LSCD no matter what caused it.
And I'm looking at table 1 in the article here, and it says they got transplants of these lab-grown corneal epithelial cell sheets. They call them ICEPS in one eye only. What did the surgery involve?
Well, they had to carefully remove the scar tissue from the cornea first, and then they stitched the ICEPS onto the affected eye. It's a very delicate procedure, and they need a highly skilled surgical team.
I bet. Did it work? Were there any improvements?
Yeah, the results are pretty exciting. After 2 years, there weren't any serious side effects reported, which is really good news. Because sometimes with stem cell therapies, there are concerns about tumor formation, but there was none of that. And all 4 patients had better vision right away, and the affected area of their cornea shrunk. That's amazing. Like, to restore vision after that much damage is incredible. Did those improvements last?
For 3 out of the 4 patients, the improvements lasted the whole 2 years they were observed. One patient did have some regression, but the overall results are still really promising. Of course, it's important to remember that this was a small study, but these initial findings are encouraging for sure.
Totally. Reading the article, though, it seems like there's some uncertainty about how exactly these transplants work. Like scientists aren't totally sure about the mechanism behind it.
Yeah.
Can you help us understand that a little?
That's a really good point. There are a couple main theories. One is that the transplanted cells just directly regenerated the cornea. The other is that the transplant acted as more of a trigger, like it stimulated the patient's remaining stem cells to repair the damage themselves.
So like it could be a direct fix or a jump start for the body to heal itself.
Exactly. This happens a lot in regenerative medicine. We see positive outcomes but aren't totally sure about the specifics of the mechanism. They'll need to do more research to figure that out.
Well, regardless of how it works, the results are really promising. This is a big step for treating LSCD. Let's zoom out a bit and talk about stem cell therapy for eye problems in general. We have that review article too. What are the key takeaways there?
Well, the review article talks about all these different types of stem cells they're exploring for ophthalmology, each with its own special properties and potential uses. We're talking embryonic stem cells, mesenchymal stem cells, retinal progenitor cells, and more.
So it's not just iPSCs. There are tons of different stem cell types that could be used to treat a bunch of eye diseases.
Yep. And they've had some pretty cool successes already treating things like cataracts, glaucoma, macular degeneration, and retinitis pigmentosa using different stem cell approaches. They've been able to slow disease progression and even improve vision in some cases.
Wow, it sounds like stem cell therapy could totally change how we approach eye diseases in the future.
It definitely has huge potential, but like any new tech, there are challenges like immune rejection potential for tumors and the need for standardized protocols.
Makes sense. We talked about immune rejection before, but that's still a concern even with iPSCs.
Well, it's less of a risk when you use a patient's own cells, but it's not completely eliminated. They're working on some strategies to address this, like creating universal donor cells that wouldn't trigger an immune response, so you wouldn't need to use immunosuppressant drugs.
That would be amazing. What about the tumor risk? How are they handling that?
So they're engineering stem cells with these built-in safety mechanisms, almost like a molecular brake that stops cell division if things start to go wrong, like a kill switch for uncontrolled growth. And they're looking into using biocompatible materials to encapsulate the stem cells, so they're kind of in a protective barrier and can't interact with surrounding tissue and form tumors.
So a multi-pronged approach to make sure it's both safe and effective.
Mm-hmm.
And what about the standardization issue? Why is that so important?
Well, right now, there's a lot of variation in how stem cells are derived, cultured, and used in trials. It makes it hard to compare results from different studies and create solid treatment guidelines. Standardized protocols are super important to make sure the results are consistent and reliable. Like, you need a good recipe to bake a cake, otherwise it won't come out the same every time.
That's a great way to put it. It sounds like there's a lot going on behind the scenes to make sure these treatments are brought to patients safely and responsibly.
Absolutely. It takes a team effort, biologists, engineers, ethicists, regulatory experts, everyone working together to make sure these therapies are developed and used with care. Yeah, it really is a huge undertaking. It's amazing to see all these people working together to move this field forward.
Yeah, for sure. It makes you think about like the bigger implications of all this. Like if scientists can grow actual working corneal tissue from stem cells, what else could be possible? Could we see other organs or tissues being regenerated?
Yeah, it's mind-blowing when you think about it. Like, zooming out to the bigger picture, this research suggests that maybe someday organ transplants, as we know them, won't even be necessary. Imagine growing a personalized organ from a patient's own cells. No need for donors, no risk of rejection.
That would be incredible. No more waiting on those transplant lists, no more worrying about finding a match. That would be a game changer for medicine.
Exactly. And it's not just about replacing whole organs either. You could use this tech to repair smaller things like a torn ligament or a damaged heart valve.
So instead of patching things up, you could actually rebuild the tissue. That's amazing.
Yeah, and it opens up all these possibilities for personalized medicine. Imagine tailoring treatments to each person's specific needs, targeting the damage with the right cells and growth factors.
That level of precision is really exciting. It makes me wonder about prevention, too. Could we use this tech to strengthen tissues before they're even damaged? Like, could we prevent injuries before they happen?
You're thinking ahead, and that's where this research is going. The goal is to be proactive about health, not just react to disease and injury.
It's about empowering people to take control of their own health. And with stem cell therapy, it's not just about living longer, it's about living better.
Exactly. It's about increasing our health span, the number of years we're healthy and active, living longer and healthier and enjoying those extra years.
That's a great goal to have. This Lancet study was about LSCD, but what about other eye problems? Could this kind of iPSC therapy work for those too?
Definitely remember that review article. It mentions a bunch of other conditions that could potentially benefit from iPSC-based therapies. Macular degeneration is one example. It's a major cause of vision loss in older adults. They're looking at using iPSC-derived cells to treat it.
It's amazing to think that stem cells could be a solution for macular degeneration. That affects millions of people.
And it's not just about restoring lost vision either. They're also looking at ways to use iPSCs to treat other parts of the eye like the retina.
The retina is so important for converting light into signals our brains can understand. If you could fix damage there, that would make a huge difference.
Exactly. And they're even looking at using iPSCs to treat glaucoma, which damages the optic nerve that carries those signals from the eye to the brain. So the potential is really broad.
It sounds like we're on the edge of a whole new era for eye health. It's pretty incredible.
It's definitely an exciting time for ophthalmology, and things are moving really fast.
You mentioned that the scene cultivation method used in this study is groundbreaking. Why is that?
So the SEAM method, it stands for Self-Formed Ectodermal Autonomous Multizone. It's basically growing iPSCs in a 3D structure that mimics how the eye develops naturally.
Oh, so it's like that mini eye we were talking about before.
Exactly. By recreating that natural developmental process in the lab, they can guide those iPSCs to turn into the specific cell types they need to treat different conditions.
That sounds super complex. What's the advantage of doing it that way compared to other ways of growing iPSCs?
One big advantage is that you get very pure populations of specific cell types. That's really important for safety and effectiveness. You want to make sure you're using the right cells in the right place.
It's all about precision. What else makes this method so good?
It's also very efficient. They can produce a lot of cells relatively quickly, and that's important for scaling things up from small research studies to clinical trials and eventually to widespread treatment.
So it's a method with both short-term and long-term potential. Are they using it to study other things besides eye diseases?
Definitely. They're exploring it in a bunch of different fields like neurology, cardiology, and even cancer research. Being able to create these specific cell types in a controlled and efficient way is a big deal for all of regenerative medicine.
Wow. It's like they've invented a whole new set of tools for scientists. It's amazing what they can do now. This has been a really fascinating deep dive. We've talked about how iPSCs and this C method could revolutionize the way we treat eye diseases and maybe even a lot of other conditions. But we've also talked about the challenges involved. Before we wrap things up, let's go back to those challenges and the things they're doing to address them.
It's important to be realistic about the challenges, even when we're excited about the progress. We talked about immune rejection and how, even though iPSCs come from the patient's own cells, there's still a chance the body might reject them.
Yeah, that's a big concern, especially if you're talking about long-term treatment.
Mm-hmm.
What are they doing to reduce that risk?
Well, one promising approach is to create universal donor cells. These would be iPSCs that are basically invisible to the immune system. They might modify the cells to remove the things that trigger the immune response, or they could use gene editing to give the iPSCs specific properties that let them evade the immune system.
That's amazing. To have stem cell therapies that are readily available and don't require immunosuppressant drugs, that would be a huge step forward.
It's a big goal, but it's definitely possible. Another challenge we talked about is the potential for tumor growth. Stem cells, by their nature, can divide and multiply very quickly, and if that's not controlled carefully, it could lead to uncontrolled growth.
That's a valid concern.
Right.
What are they doing to prevent that?
They're developing really sophisticated ways to engineer stem cells with built-in safety mechanisms, like molecular brakes that stop the cells from dividing if things start to go wrong.
Like a kill switch for out-of-control cell growth.
Exactly. And they're also looking at using biocompatible materials to encapsulate the stem cells, which creates a barrier that prevents them from interacting with the surrounding tissue and potentially forming tumors.
So a multi-layered approach to make sure it's safe and effective. We also discussed the importance of standardizing things in this field. Can you remind us why that's so important?
Right now, there's a lot of variation in how stem cells are made, grown, and used in clinical trials, and that makes it hard to compare the results from different studies and come up with clear treatment guidelines. Having standardized protocols is essential to make sure the results are consistent and reliable. It's like having a recipe for baking a cake. You need to follow the recipe if you want it to turn out the same way every time.
That's a great analogy. It sounds like there's a lot of work going on to make sure these treatments are safe and effective for patients.
Yeah, it takes a lot of people working together to get this right, and it's all about helping people in the end.
It's really amazing to see how far this field has come. This deep dive has been full of exciting discoveries. We've learned about iPSCs, the seam cultivation method, and those incredible results from the first corneal transplants using iPSCs.
It's been quite a journey, hasn't it? We've talked about everything from stem cell basics to the idea of regenerating organs. It's mind-boggling.
We've also been realistic about the challenges and all the work that's being done to overcome them. But before we wrap up, I want to talk about something we haven't touched on much yet, the human element of all of this.
I think that's so important. Every scientific breakthrough has a human story behind it, stories of hope and resilience.
Right. So I want to leave our listeners with something to think about. Imagine you're one of the patients in this study. You've been dealing with vision loss for years, and suddenly you have a chance to see clearly again. What would that mean for you? How would it change your life, your relationships, your whole sense of self?
That's a really powerful question. It reminds us that science isn't just about numbers and data. It's about people. It's about improving lives and giving people hope.
This deep dive has shown us that regenerative medicine could completely change how we treat diseases and injuries in the future. It could give new hope to millions of people around the world. It's been an amazing conversation, and I hope our listeners have enjoyed it.
I know I have. It's always great to talk about these breakthroughs with someone who's as excited about the possibilities as I am.
And to our listeners, thanks for joining us on this deep dive. Keep exploring, keep learning, and until next time, keep your eyes on the future